Okay. Good evening, everybody. Welcome to the week 9 of the course, Enzyme Science and Technology. We will wait for some time for everybody to join in. After that, we can be beginning with today's session. Let's wait for some time, let everybody join, and then we will start. So I'll just keep the camera off for a while, and then we will begin. Yeah, okay. Good evening, everybody. Let's begin with the session. As usual, we will be starting with a certain recap of whatever has been taught this week, and then we will solve some questions. And today's topic is about inhibitor design. So let's see what it is all about. Inhibitor design. So we'll talk about inhibitor kinetics. So talking about inhibitors, they are specific for enzymes. They can be from natural sources, like they can be from plants, or they can be from other organisms, right? They are synthetic chemicals. They disrupt the enzyme structure. And we will see how that happens, right? They reduce the enzyme activity. Also, they are modifiers because they alter the specificity of the enzyme towards the substrate. So that's the idea. So let's say, for example, if you use urea or guanidine HCl, okay, guanidine HCl or beta mocaptoethanol, they all denature enzymes, right? They are denaturants. Uh, and beta amine specifically targets the sulfhydryl bonds. Sorry, there's some sort of problem. Just give me a moment. Right? It breaks the sulfhydryl bonds. There could be other things like DTT as well or CTEP, which can be used as well for reducing these bonds, right? Now, um, the enzyme structure determines whether there would be the binding of substrate or inhibitors, right? We know this. And the binding pocket, let's just take this. The binding pocket is called the active site. Although there could be other sites of binding pockets, which are others. There could be allosteric regulations and things like that. So for designing for substrate or inhibitors in specific, we have to design the molecule and we can actually generate a library very similar to cDNA library, which we have already discussed. Similarly, we can generate a library for enzyme inhibitors, right? So for understanding how the mechanism of enzyme action, we have the Fisher's lock and key model. Fisher's lock and key model. And it was introduced by Emil Fisher in 1898 and still very much accepted. Actually, what it says is that the enzyme and substrate are specific to each other, just like a lock and a key. So if you see the enzyme, let's say this is the enzyme, This is the structure of the enzyme. And there is a substrate attached to it. This is the substrate. It forms something called the enzyme substrate complex. Okay. So what are the criteria here which are important for you to understand? First is there is a reciprocal fit between enzyme and substrate, right? Here, two molecules of are involved. Two molecules are involved. So the concept of intramolecular fit is basically what we have, right? This complex is highly unstable and it decomposes to produce enzyme plus product. So how it looks like is the substrate will actually decompose to form the enzyme will be back in its reusable form. And what we get out of it is a product. So this is the product. So this enzyme substrate union causes release of energy. Okay? So it causes release of energy and the substrate reaches an activated state because of this energy. It reaches an activated state and so substrate reaches an activated state, and in this activated state the substrate is more susceptible to cleavage. And that is how the product is formed. One of the major limitations of this model is that it assumes rigidity as a factor. So it does not account for that enzymes can be not rigid, they can be flexible as well. So that is not taken into account in this model. And to overcome that, there is another model. Wait, just give me. There's another model that is the uh, Koshland's induced fit model that overcomes this, this limitation. So it presumes that enzyme does not retain its original shape and structure. But what happens is contact of substrate induces configurational or geometric uh, changes, right? So what happens as a consequence is that enzyme fits completely. I'll show a diagram and it will make it. Uh, it will be more clear in this way. So what happens is, let's say this is the enzyme, and the enzyme has a structure like this. Let's say this is the enzyme, right? And there could be different substrates. Substrates could be like this, and they could be like this. They have a different structural conformation. What happens is the enzyme retains the shape of the substrate. This is how the enzyme looks like. Retains the shape of the substrate. This is the substrate here, and this is the substrate here, right? So let's just shade it, and it retains the size, and then it forms the product. So the enzyme, and now the product is formed. And then it will lead to release of these products, and the enzyme can again be reused, right? So this is enzyme and substrate. This is the enzyme substrate complex. This is the enzyme product complex, both of which are intermediary stages, not very stable. Now, what are the three possibilities that is happening among the enzyme that makes it bind to the substrate? Okay, there could be three possibilities. One is enzyme may undergo conformational change and then bind to substrate. Okay, what else can happen? Substrate may bind first, induce conformational change, in the enzyme. That can happen. Or what else can happen? So both processes may happen simultaneously. ठीक है? Leading to further isomerization, final conformation. The Koshlin's model also known as the induced fit model, gained more popularity. Okay, now let's talk about inhibitor design, right? Let's do this. This is very important. Koshland's model is also known as the induced fit model. I think I should have made it a little bit more lighter. But it's fine. Now talking about the inhibitor design, right? So the enzyme structure as small areas of this enzyme are active sites. Okay, it's part of subset of enzyme is an active site. So they have, let's say, the binding pocket, right? They have the binding pocket where they can have the active site, they can have the substrate binding site, or they can have other sites. Now let's talk about active site. Now the substrate molecule is comparatively or the enzyme molecule. Specific region of enzyme bind to the substrate. These specific regions are called the active site or the catalytic site or the substrate site. Now let's talk about the properties of these sites. Active site. Okay. So if we talk about the properties, it occupies relatively small portion of enzyme molecule. It's a 3D entity. For example, lysozyme, it has 6 subsites. The arrangement of atoms is well defined. So that gives you the specificity towards the substrate, right? That is the reason for specificity. The binding between substrate and enzyme is relatively weak. Okay. So these are actually grooves or crevices which are important for substrate to come and bind to the enzyme. Now, how to determine the active site? So active site determination can be done using 3 methods. Fine. There could be biochemical approaches which involve some wet lab work. Which will actually, you will see the substrate binding in that case. There could be bioinformatic approaches where you actually perform, things like there is for servers or databases like PFAM, which can predict the active site of a protein. And then there are structural approaches to it as well. So in this case, you can go for, let's say, docking and stuff like that. So to see what might be a potential active site, or you can also compare it with other active sites which are there, right? Now, talking about the biochemical approaches, it can be through amino acid modifiers, amino acid modifiers, or through mutagenesis. So mutagenesis can be again site directed, where you change a specific amino acid or nucleotide, or they could be random as well. Now, talking about the bioinformatic approaches, there is a database called PFAM where you can actually get about active site prediction. Now, it will basically, color the residues as red, the residues which are um, potential active sites. Now, talking about the structural approaches, we have again databases which have 3D folds. Now, talking about the inhibitor design, there could be the conventional or traditional approach. There could be ligand-based approach, and we will discuss about these, and then we will be done with the not charged. Just give me a moment to charge this. Okay, and then there could be things like sure, okay. Receptor fitting, that is done using molecular docking. We'll be talking about it. And then there is the receptor-based approach. Okay, these are all the things. Now, talking about the conventional approach, there is no need for, no information on enzyme and inhibitor structure. You have to perform enzyme activity assay to see if the activity is reduced. Then we screen the active molecules. Then we perform validation by binding assays. So for example, ITC is an example, SPR is another example. Off target, there is no information on this off targets, right? Then there could be targeted approach as well. Here the info on enzyme and inhibitor structure is known. Activity assay is not required. Okay. Here we perform virtual screening of active molecules. It's an interaction study. So in the traditional approach, for example, we have things like bioactive phytochemicals. They inhibit malaria, parasite propagation. Now, behind the rational inhibitor design, we need to know the 3D structure of the biological target. The structure of known active small molecules. This is called pharmacophore-based drug design. Then there is computer-assisted drug design. Computer-aided drug design. We have molecular graphics, pattern recognition, and then receptor fit. Okay. So now talking about a little bit about the structure-based drug design. And these drugs are essentially inhibitors for enzymes. Um, we can look at the ligand database or the target protein, which is again from a database you will get. And you will perform something called molecular docking. Then the ligand docks to protein's active site. And then we optimize the pharmacokinetics and pharmacodynamics. So there are certain docking programs. The most common one is Autodock. Then we have Gold, which is basically genetic optimization for ligand docking. Ludi and Hex, even Haddock for protein-protein docking is used. So molecular docking is the rational design of drugs. We use molecular docking. Now, it can be used to predict binding affinity using a scoring function. Okay. Can be used to identify ligands correct binding geometry. Now, if we look at the types of docking, it can be of two types. One is the rigid docking. Another one is flexible docking. Flexible docking is more computationally intensive because the residues are flexible, they can move. Rigid docking is based on the lock and key model, and flexible docking is based on the induced fit model. Okay. So docking can be of multiple types. So they can be protein ligand docking, which is the most common one. It can be protein-protein docking. Or it can be protein nucleic acid docking. Which could be DNA, RNA, anything, right? So these are the thing. If we look at the docking workflow, how will it look like? So target selection, leading to target preparation. And ligand selection, then we will have ligand preparation. So you need to prepare the ligands because, usually ligands and proteins, they do not have hydrogen bonds in them. You have to remove some water molecules, stuff like that. And then you would prefer or perform the docking. And you have to evaluate the docking results. Okay. If we look at the uses of docking, then it can be used for virtual screening. Oh, sorry. Drug targets. To understand the protein ligand interaction. To understand the machinery of life. So let's say enzyme inhibitor class, antibody antigen class, and there are others. Okay. Then for protein therapy and engineered protein enzyme. Okay. If we look at some applications, then for virtual screening, so without performing experiments, you can screen for multiple drugs. So you can get hits basically based on that. So it's because it has a scoring function. It can be used for drug discovery based on the same principle, and also for bioremediation. So bioremediation is how certain enzymes they can degrade pollutants. You can actually screen that as well. For bioremediation also it can be used. So with that, I think we are done with the recap part. We will quickly move to the questions. Um, so talking about question one, allosteric inhibition is a kind of competitive, non-competitive, suicidal, or uncompetitive inhibition. So allosteric inhibition is inhibition which happens not in the active site. So let's say this is the active site, and this is the active site, and the inhibitor binds somewhere here, not at the active site, some at an allosteric regulatory or allosteric site. Okay. This kind of inhibition is a non-competitive inhibition, does not compete for the substrate binding. That's the main thing. Inhibition of invertase by sucrose is a type of dash inhibition. Is dash type of inhibition? Is it substrate inhibition, non-competitive inhibition, product inhibition, or competitive inhibition? It's an example of substrate inhibition. Okay. Match the inhibitors of the protein and enzyme with its target disease. So let's say A is human venin. B is collagenase. C is purine nucleotide phosphorylase. And D is thymidylate synthase. Thymidylate. A is, okay, one is antihypertension. Two is anti-cancer. Three is antidepressant. And fourth is anti-proliferation. So renin is anti-hypertension, right? Uh, Collagenase is anti-cancer. Purine nucleotide phosphorylase is antidepressant. And thymidylate synthase is anti-proliferation. So A1, B2, C3, D4 would be the answer. The Autodoc, in Autodoc, the term grid refers to graphical user interface, docking score, 3D spacing around the target proteins where docking is performed, or the ligand file. So it's actually the 3D spacing of the target protein surrounding the ligand file where docking is performed. Basically, that is where it is. In drug design, targeting the active site of proteins is important because it disrupts the protein secondary structure. It allows specific interaction with the substrate molecules. The active site is less important for protein functions. It only affects enzymatic catalytic activity. So active site allows specific interaction with the substrate molecules would be the answer. The term pharmacophore in inhibitor drug design refers to the active site of enzyme, class of reversible inhibitors, mechanism of Mechanism-based inhibitor, essential features of drugs that contribute to its biological activity. So it's the essential features of drug that contributes to its biological activity. That would be the answer. Suicidal inhibition, also known as mechanism-based inhibition, involves irreversible inhibition of enzyme, reversible inhibition of enzyme, binding of inhibitor to active site, and activation of enzyme. So it means irreversible inhibition of enzyme. The main information obtained from NMR spectra in structural biology is the mass of the molecule, the three-dimensional structure, absorption of visible light, or the surface area. So we know NMR is for the 3D structure of the molecule. Initial velocity of enzyme catalyzed reaction is 4 micromole per minute, and the substrate concentration is 2 millimolar. If enzyme concentration is 0.5 millimolar, calculate Michaelis-Menten constant. So for this, we need to have the, give me a moment. V naught is equal to V max S by Km plus S, right? So, so this is given. V max substrate concentration is 0.5 micromolars. We'll come back to it. It would be 0.5 millimolars. The receptor-based drug design, the term allosteric inhibition refers to inhibition of an active site, uh, inhibition of binding to a site other than active site, inhibition of drug absorption, or inhibition of drug metabolism. So inhibition of binding to a site other than the active site would be the answer. The process of designing inhibitors that mimic the structure of natural ligand for target receptors is reverse pharmacology, structure-based drug design, ligand-based drug design, or de novo drug design. So it would be structure-based drug design. For an enzyme catalyzed reaction, again, this is given. Calculate the slope and y-intercept. So the slope would be 1 by V max, and the y-intercept would be Km by V max. The scoring function in molecular docking refers to determining the similarity between ligands, evaluating the fitness of ligand conformation in binding site, calculating the molecular weight, or estimating the melting temperature of ligand. Evaluating the fitness of ligand conformation in binding site. That would be the answer. In uncompetitive inhibition, the presence of inhibitor affects Vmax and Km. In which way? So it actually, the Km, it decreases and the Vmax increases. Software uses for molecular docking studies is Photoshop, AutoCAD, PyMol, or Autodoc. It would be Autodoc. Autodoc is the answer. So with this, we are done for today's session. We'll be meeting again next week at the same time, 6 to 8 p.m. for the next session for week 10. And we only have 3 more weeks left. So I wish you guys a very good luck in solving the assignments. With your permission, I would like to leave the meeting right now.